Hey guys, so now I'm gonna just answer some questions. This video is to answer some questions from the puzzle videos when I ask like, "Hey, do you guys have any questions?" so far? So there's quite a bit of question. People ask quite a bit of questions. It's good to ask these questions. So I'm just gonna make a video just to explain these things. So how specific does specific immunity get? So are there any specific immunities that can target multiple? Um, that's a good question. Now, usually when you talk about specific immunity, you're talking about an antibody specific to one type of antigen. Now, are there possibilities that you can target multiple pathogens because they have similar enough antigens? That's a good one. That's the second question. That's a good one. That is a potential, considering the fact that that is a potential because a lot of times these antibodies specific to a certain type of antigen, if the structure is close enough, it can potentially trigger an immune response. Now, can that happen? I'm not sure, but theoretically, it is possible. Okay, theoretically, it is possible. Um, Now, actually, you know what? For example, influenza virus. Okay, influenza virus. So a lot of times you have this H1N1, H5N1, H blah 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 blah. Now the thing is this: if H1N1 basically talking about the two, there are more than one, more than two antigen, but there are two antigens on the influenza virus. So if you think about it, H1N1 and H5N1 influenza virus will share the same antigen, which is the N1 antigen, and something anticoagulant. I have to look that up. So in this case, if there is an antibody for that particular antigen, probably, yeah, probably there will be confer certain level of immunity. However, would there be one? I'm not sure. Okay. So it is a theoretical possible question. That's a very good question. Now, does non-specific immune response from macrophage bind occur randomly? More or less, yes. So we have tons of those guys floating around, leukocytes floating around to check on these things. Like, do antibodies have to check molecules in the bloodstream? Yeah, they take time. Does the specific take longer? Partially because I had to prioritize. Um, prioritize also if you look at the diagram. So in this case, specific immune response take longer because you have to kind of, part of it, you do wait for the phagocytes to take out some of the antigen and present that. And then you also have other middle sites here, which can take up quite a bit of time. So in this case, the process does take longer. And then you also have to find one that is appropriate to the B cell, appropriate B cell for that particular antigen. So it does take longer. Now, I was looking at the higher level material, and I was just say right now, higher level students, we will go over this process in a much more finer detail. Much more detail, so it will help you to understand this process better. Standard level students, this is it. This is actually these 5-6 steps. That's pretty much as deep as you need to know this part. Okay, so specific immune response does take longer because you have to go through more steps. If it is a secondary response, i.e., you've seen this virus or this bacteria before, and now your memory B cell kicks in, that will just be so much faster. Okay, response will also be so much stronger too. If lymphocytes are programmed to attacks, yes. Yes, potentially yes. Or are these very avoid detection? So that, yeah, that too. Both of them are true. Now, can CRISPR This is a part where I'm not familiar with CRISPR as much, simply because um, it's never been an IB syllabus thing. But also another thing is that there has been in the past, at least when I was a student, studies of can you use your own body immune cells to attack cancer. Now, how far has that research gone to, or have they continued with that approach? I don't know, but that's something worth looking. But cancer cells do come up with ways to avoid detection, i.e., they try to they partly it is because it is their own. You're talking about your own body cell, right? Immune cells usually attack foreign cells. Cancers are technically your own body cells, but just gone haywire. So yes, there are still certain cancerous cell markers on the surface of the cell that immune cells can look for that, but otherwise it can be pretty tough. So those are good. That's a good question too. Handle down page 3. There is a diagram in green antibodies and red pathogen with is a square labeled different antibodies made for this. What does that mean exactly? So let me take a look at this. Yeah, that's a good question. So I think some people ask me this question during office hour or during a lesson. So in this case, yes. So if you look at it, this antibody is specific to this particular antigen because structurally they are similar. Okay, structurally they are compatible or similar. It's very much like the enzyme and the active site and substrate thing. So the structure is compatible. So this antibody will be specific to this particular antigen. You need a different antibody to be specific for this antigen itself. So that's I think that's a good thing to ask that question. Do you have to create and label a diagram of this? I presume that's a question. My answer is no. It certainly can help, but you don't need to be able to draw this. Main difference between lymphocytes and leukocytes, helper T cell lymphocytes. Okay, now they're both white blood cells. Leukocytes are generally non-specific immune response. Lymphocytes are specific immune response. Helper T cells are lymphocytes. So both T cells and B cells are lymphocytes, this kind, okay? Antigen on the phagocyte surface bind to the specific response T cells. Helper T cell, this similar action occur also. Yes, they both happen to that. You said it's important to note that antibody is specific. Okay, what do you mean by that? When you say complementary, do you mean an enzyme? Yes. So these two, I think I answered the question already. It is specific to one type of antigen. So one type of antibody is only specific or will bind to one type of antigen. It's kind of similar. So it's very similar to what we were talking about, enzyme substrate complex. They're not enzymes nor substrate, but they are following that same concept, that same idea. Okay, so structurally, one has a particular shape and the other one has a compatible or complementary shape which binds to it. Okay, are there specific antibodies? Yeah, I think that's what I was talking about just now. What are virgin B cells? Virgin B cells are the B cells which have no, they are not memory B cells, so they're not, they have not previously met a particular kind of antigen. In fact, on this outline here, now it's not something you guys need to learn, I believe. Where is it? Oh yeah, Wikipedia. Yep. I know it's not a scholarly site, but they do have a very good diagram showing. Now, partly now clonal selection. I believe this is partly the higher level material. However, keep in mind, keep in mind, this stuff that here it's pretty much third year university and fourth year university stuff. So what I'm teaching right now, it's kind of a simplified version and some basic stuff. Okay, so when you're talking about clonal selection, so in this diagram, so if you look here, you have stem cells turning into different kinds of immature lymphocytes, and we are only really focusing on B cells right now. Now, there are two kinds of selections. There's some kind which is what they called, forgot the name of it. Is it negative selection? Yeah, negative selection. Just double checking. Negative selection, negative selection. Okay. I can't find it right now. But basically, now, so these stem cells made a whole bunch of different kinds of lymphocytes. And then if you click on this diagram and made it bigger, you notice that all these lymphocytes, these B cells, have different kinds of antibodies. Okay, now this part here is essentially now because our body have many different types of cells, right? And our body on in one way for your immune cells to make sure that they know whether a cell that they encounter it's from your own body or from a foreign cell. So one of the things they do is that in the bone marrow, while these immature lymphocytes, B lymphocytes are being formed, they will be exposed to a whole bunch of antigens that are from your own cell body. Okay, this is from your own self. So any lymphocytes that will be specific against your own self antigen will be eliminated. So those will be selected against. It's a mechanism to kind of protect yourself from autoimmune disease. So imagine if these B cells somehow miss this quality control, this selection process, you'll have B cells which will create antibodies against your own cell, and that's a bad thing. Okay, so this part is to protect yourself. Now you have these here. These are also virgin B cells. Okay, so these are virgin B cells. Now in this case, number 5, you have a whole bunch of foreign antigens coming in. So when you have these whole bunch of foreign antigens coming in, out of these 3, there are of course many more, 3 virgin B cells, only this one is compatible or complement to these new antigens. So in this case, this part here, so this particular one will be activated to multiply. Okay, so these becomes activated. Some of them are going to become plasma cells. They are going to keep churning out antibodies, but they only will survive or live in this system for a few days. Whereas, let's just say some will become memory B cells, which will live in your system for decades at a time. Okay, so this is actually a theory called clonal selection theory. Okay, this is some of it you'll see in the higher level material, but a lot of this might be just way over in the university. Um, someone was asking about, people were asking about this, the relationship between virgin B cell and how this whole thing works. So I decided to look this up to explain, to help you explain it. Hopefully, this makes things a lot more clear. Yeah. Any questions? All right, so let's go back to that list of questions that people have. Yeah, I think I talked about this already. Only B is complement. What is this complement? Yeah, it's very much like lock and key idea. Is there any research possibly accelerating process that specific immune undergoes to fend off new disease? Not really, because if you want to speed that process up, you never really quite know how these quality control issues might work. And in fact, a lot of times we are focusing on toning down the immune response. So, for example, transplant, or even the case of things like allergy. You're trying to tone down your immune response to allergy. Now, we're going to talk about allergy if you take HL. Okay. Yeah, we're going to be talking about these other components in today's class and maybe one more class. What do we need to know about virgin B cells? I think I answered that question earlier just now. Why can't phagocytes stimulate the B cells on its own? They just don't. They just don't. You just don't. They're not really the ones that sends out signals. They are presenting the antigen, but mechanism-wise, they're not the one presenting the signal. It is T cells that is necessary. In fact, the helper T cells are necessary because they are actually part. Now there's a heck a lot more of this stuff, but they are the ones that will help activate the antibody being formed. So in this case, helper T cell is very necessary. And in fact, HIV virus tends to attack helper T cells. And in this case, if you think, whoa, how immunocompromised HIV positive or people with AIDS are, you can see like how important helper T cells are. Okay, so I hope this video helps to answer these questions. Again, immune system is a tricky or denser topic, so feel free to ask these questions on the puzzle question part or whatnot. Okay, I'm very glad to see that you guys are asking that, and I'm gonna make the second video for tomorrow's lesson now.